咁啊成件事都俾你搞彎晒啦，係咪？
俾你搞彎晒啦！
喂，咁多位大家好，我係梁 Sir。今次呢段片呢，我哋就嚟講下分子遺傳學同埋癌症嘅。今次呢，我哋就要介紹一款致癌嘅化學物質，就叫做 BAP 或者 BAP 啦。咁啊喺一啲燒肉啊、烤肉呢嗰度揾翻嚟嘅，即係一啲經過高溫烹調嘅時候呢，就有機會釋放出嚟嘅一款化學物質啦。咁佢呢，就能夠黐喺我哋嘅 DNA 嘅 nucleotide 上面嘅。咁啊隨機地黐啦，咁如果佢係黐咗落去個 guanine 個 G 嘅時候呢，咁佢就會被錯誤讀咗為 T 塊面噶啦。咁啊題目俾咗個好明確嘅指示你嘅，咁呢個係我哋嘅 DNA 啦，個雙條啦，跟住啦個 BAP 就黐咗落嚟啦，咁啊咁啱得咁巧真係黐咗落個 G 度喎。咁 Part A 呢就問啦。根據翻呢一幅嘅圖，咁我哋寫翻咧，我哋個 nucleotide 嘅 sequence 嗰個核苷酸嘅序列，咁啊喺翻個 opposition， 即係話啦，喺對面呢一條。如果個錯讀真係發生咗嘅時候，咁其實呢條題目呢，就係考翻我哋 ATGC 個配對啦。今年嘅 MC 其實都係考類似嘅嘢嘅。我知啦，互補鹼基呢，大家一定要學識咯。咁嗱，前面呢 AAC 上面配 TTG 就冇得走雞噶啦。但係而家呢個 G 呢，就被錯誤讀為 T， 咁所以對面呢就會變成 A， 咁所以答案呢就係 TTGA 啦。咁呢條題目啦，有咩嘅變奏呢？今次就講啦，下面呢 AAC 就誤讀咗變做 T 啦，可能話俾你聽啦，呢一條呢可能係 template strand 啊樣板鏈，又或者 coding strand 密碼鏈。跟住啦，究竟個 mRNA 啊信使 RNA 個 base sequence 會係如何呢？又或者啦個氨基酸嘅序列又會係如何呢？有心理準備，人哋會咁樣問你咯。跟住啦去到 Part B 啦，佢就問啦，講一個理由就解釋下呢一類型嘅突變，即係話啦呢個 G 俾人錯讀成為咗 T 呢，係可能係唔會影響到所生成嘅蛋白質嘅功能。咁考緊我哋啲乜嘢呢？第一啦，就係呢一款嘅突變究竟係邊一款呢？就係將個 G 誤讀變成 T， 佢就好似一個取代啦 ，substitution 啦。第二啦，一個蛋白質嘅功能，其實我哋係點樣控制佢嘅呢？咁啊，過往我哋都有類似嘅題目嘅，其實就係有關於個蛋白質嘅 conformation 個構象啊，即係佢個形態形狀啦，係咪？咁究竟佢個氨基酸嘅次序係如何啊？佢個構象啦，點樣翹埋嘅呢？單條變做螺旋，由螺旋變做立體，由立體一嚿蛋白質楞住另一嚿蛋白質，會唔會咁樣先帶得出個用處呢？第三啦，就係考翻我哋有關於 genetic code 嗰個 degenerate 簡拼嘅特性啦。咩叫簡拼啊？就係唔同類型嘅密碼子，原來啦佢哋所去對應嘅氨基酸呢都係一樣嘅。嚟啦，睇翻呢個密碼子嘅表格啦 ，UUU、UUC， 原來啦所對應嘅氨基酸呢都係呢款 PHE， 呢個就叫簡拼啦。所以 Part B 最緊要嘅概念就係。雖然而家我哋發現有基因嘅突變，但係如果啦佢係冇改變到個蛋白質嘅構象嘅話，例如啦冇影響到個酶嘅活性位置嘅形狀，咁呢一個酶嘅分子或者呢一個蛋白質仍然能夠正常運作嘅。咁所以 Part B 呢，其實喺個技巧上面就考翻我哋個因果嘅關係啦。個因就係呢一款嘅突變係取代。而個結果就係蛋白質功能，冇變喎。所以新嘅三元密碼子呢，其實佢哋都係對應翻同一款嘅氨基酸。又或者啦，雖然突變真係發生咗，佢真係整咗一個新嘅氨基酸，但係呢個氨基酸係冇影響到個蛋白質嘅功能。例如啦，佢唔係喺個酶嘅分子嘅活性位置嗰處，咁所以佢咪唔會影響整嚿酶嘅功能咯。咁呢兩個呢，都係可能嘅原因嚟嘅。咁啊，講翻少少常犯錯誤啦。咁有啲同學咧就純粹就係講啦，密碼子咧係簡拼噶，所以就冇事啦。咁其實佢哋就係冇咗個解釋咯。成日話直線抽擊答題法，我哋要留意嘅自己係火車到咗站未。如果你淨係講佢係簡拼嘅密碼子，咁火車係未到站。你要講下啦，簡拼咗之後，原來呢就係不同類型嘅密碼子都係對應去翻譯翻同一款嘅氨基酸，咁咪唔影響佢嘅構象、佢嘅功能咯。同埋啦，有啲同學呢 就過分簡化咗個概念，就以為啦一個突變咗嘅密碼紙，佢係去整同一款嘅蛋白質，但係其實個密碼紙係攞嚟對應翻相同嘅氨基酸啫嘛。佢哋就簡化到係啊，因為咧即使係突變咗，都係整咗同一款嘅蛋白質，就當答咗題目啦。其實係個概念唔清晰就真。跟住去到柏斯啦，佢就話呢一款嘅突變，隨住時間過去，繼續積聚喺個 DNA 嘅分子當中，咁就真係有機會導致到嗰個蛋白質嘅功能就受到影響啦。咁你諗下嗱，而家呢一個 BAP 咧，係隨機地黐落去個 DNA 個樹啊嘛，黐落個 G 嗰度啊嘛。咁你諗下成個 DNA， 咁你有好多個 G 噶嘛，係咪？咁全部錯讀晒變做 T 嘅話啦，咁當你去整嗰個 mRNA， 跟住嗱 codon 又俾你整彎咗，咁啊成件事都俾你搞彎晒啦，係咪？咁呢條題目其實考緊我哋嘅就係究竟個細胞嘅咩嘅過程，當佢失控嘅時候就會導致到腫瘤，甚至乎係癌症呢。嗱，今次講腫瘤啦，佢再推多一步，咪可以問癌症咯。咁整個思考邏輯就係講緊啦，個基因佢控制我哋整某一款嘅蛋白質，而呢款蛋白質就控制某一個細胞嘅過程，而其實呢個細胞嘅過程正正就係細胞分裂。咁其實我哋嘅細胞分裂咧就係由個基因所去控制啦。咁如果呢一個基因係有基因突變嘅時候啦，咁即係我哋所產生出嚟嘅蛋白質都有機會係功能不良，係嘛，做唔到正常嘅嘢。咁我哋呢一個嘅細胞過程，其實即係細胞分裂啦，就會受到過分嘅刺激，又或者唔識得抑制咗佢，咁就會導致到腫瘤嘅形成啦。咁所以答案呢都不難理解啦，就係呢一款蛋白質係控制細胞分裂啦，控制有絲嘅細胞分裂啦，或者控制個細胞周期啦，諸如此類嘅，又或者佢係控制緊個 DNA 嘅修復啦。其實我哋人體係有能力去修復個 DNA 嘅。好，咁我哋嚟到一點出發啦。咁今次呢條題目就由鹼基序列開始啦，然後啦就問翻製造蛋白質啦，同埋有關突變嘅概念啦。咁啊，首先啊，先講誘變劑，就係呢一款致癌化學物質啦，就係咧由呢啲烤肉呢咁高溫烹調嘅嘢食咧，所以產生出嚟嘅。咁下次咪可能問下你咯，喂，其實咧，如果我哋建議啲人去烚啲嘢食，去蒸啲嘢食，其實啦係能夠減低到患有癌症啊、腫瘤嘅機會嘅，你有冇得解釋一下啊？咁梗係可以啦，就係因為啦，烤肉啊、焗爐啊、氣炸鍋啊諸如此類，極高溫去煮啲嘢食嘅，就有機會令到一啲可能加工過嘅嘢食啦，就產生呢啲致癌嘅物質。咁啊，提升咗基因突變嘅機率啦，係咪？跟住啦，就有關於鹼基序列同突變嘅關係啦。咁其中一個概念就係基因突變啦。咁啊，唔同嘅基因突變，咁啊自己溫翻書咯。幾時係一個刪減啊、插入啊、取代啊、倒轉啊，諸如此類啦。咁啊，除咗基因突變，咁當然仲有染色體嘅突變啦，例如啦唐氏綜合症啊，多咗一條少咗一條嘅性染色體啊，諸如此類啦。咁有關於製造蛋白質，轉譯轉錄冇得走雞噶啦，密碼紙嘅特徵啦，今次咧就考咗簡拼。咁除咗簡拼之外啦，仲有點啊？三個一齊行嘅，之間咧係唔會有重疊嘅，同埋啦佢哋係通用嘅，即係話啦，無論你係烏蠅啊、香蕉啊、貓啊、狗啊、人啊，對於 AAA 嘅呢一個密碼紙咧，我哋都係去整同一款嘅氨基酸嘅。然後題目啦就梗係講腫瘤啦，要考我哋嘅就係有關於新陳代謝嘅活動啦。究竟個基因點樣製造蛋白質，控制新陳代謝嘅活動，或者控制細胞嘅活動，同我哋嘅腫瘤啊、癌症啊有啲咩嘅關係呢？咁今次要考嘅當然係細胞分裂啦，亦都考下有關於細胞周期嘅概念啦。咁啊，再推而廣之嘅就係有關於良性腫瘤同埋惡性腫瘤嘅分別啦。咁過往啦都有題目係關於突變啊、癌症嘅，咁啊快啲睇下片溫下書咯。最後尾啦又講下啲題型嘅變奏啦。究竟啦，刪除同埋插入一個鹼基啫，究竟會唔會導致到氨基酸嘅改變呢？咁啊，解釋下你嘅答案啦。嗱，是但一個啦，即係你睇下，刪除一個鹼基啊，或者插入一個鹼基啊，會唔會導致呢件事啦？而第二呢，就係關於腫瘤咯。咁我哋頭先啱啱講過啦，腫瘤細胞之所以出現，就係因為我哋嘅細胞不斷去做細胞分裂。 咁究竟呢個不斷製造細胞分裂嘅意思係講緊我哋個間期係縮短咗啊，定還是我哋嘅有絲細胞分裂係加速咗呢？即係話個細胞 hea 嘅時間短咗，咁呢個就係間期短咗嘅意思啦。嗰啦就係有絲細胞分裂，即係話嗰個 prophase 啊、metaphase、anaphase、telophase 呢個前中後末呢4期係加速不斷咁搣咧。你試下留低你嘅答案，睇下你 concept 清唔清？Hello student, I'm Mr. Leung. So this video we are going to talk about the molecular genetics and the cancer and tumor. So we are talking about the carcinogenic chemical BAP, which is commonly found in the grilled meat. So we use very high temperature to cook the meat. So during this process, there will be carcinogenic chemicals produced. So this carcinogenic chemical, it can attach randomly to the nucleotides of the DNA molecules. And when it is attached to the guanine, and this G will be misread as thymine T. So we have this information first, and then we have this diagram showing the double-strand DNA. So we have AACG. Now the base guanine is binded with the BAP. So for part A, on the above diagram, write down the nucleotide sequence found in the opposite strand of the DNA when misreading happens. So we have this DNA strand, and then we are going to write down the base sequence of the upper DNA strand. So for this question, it's checking us the concept about the complementary base pairing. So we can see that AAC surely for the base pairing, TTG. And what about the guanine? It's binded with the BAP, therefore it will be misread as thymine. So the final base will be A. So the answer will be TTGA. So maybe next time the possible question variation will be that based on the mutated DNA strand, it may be given that is the template strand or the coding strand. So the question will ask you what will be the mRNA messenger RNA base sequence. Or it can ask you the amino acid sequence coded in the polypeptide chain. So you need to recall the idea of the transcription and also the translation. And for part B, suggest one reason why this type of mutation may not affect the functioning of the protein formed. So for this question, the first idea we need to identify is the type of the mutation. So according to this diagram, we know that when the guanine is attached by the BAP, it will be misread as the thymine. So it looks like the substitution, right? And then secondly, we need to realize how is the functioning of the protein being controlled. So the basic idea is that about the folding or the conformation of the polypeptide chain. How is it folded from linear form to the helical form to the three-dimensional form or two protein bind together? So it's about the conformation. And the next idea is about the degenerate feature of the genetic code. It means that some codes, they code for the same amino acid. It's just like that you can recall the codon table for the CCU, CCC, CCA, and CCG. They all code for the proline, PRO, this kind of amino acid. So for the critical concept is that although there is gene mutation, may not affect the conformation of the protein molecules. For example, the shape of the active site of the enzyme molecule, so the protein can still function normally. So after we talk about the concept checking, so what about part B, any skills checking? So for part B, we need to explain the cause and effect relationship. So the cause is the mutation, which is the substitution, and then the effect is the protein function is not affected. So how could it be possible? Maybe the new trilab code, that means the codon, encoded the same amino acid as the original amino acid. Or a different codon, they still code for the same amino acid. Or even there is really a new amino acid coded because the codon is changed, so there may be a new amino acid translated, coded. However, the new amino acid is located in a region which does not affect the functional part of the protein. For example, the amino acid is not located in the region of the active site of the enzyme molecules. Therefore, the shape of the active site is still normal, so the enzyme molecule can work normally. I would like to remind some common mistake. Some students may just mention the idea of degenerate codon. However, they do not do any explanation. So as just what I say, that straight to the point, you need to be very aware about your answer. Is it enough to talk about degenerate codon? So you need to recall the concept that some trilap codon encode the same amino acid or do not change the conformation of the protein. And some students, they oversimplify or misunderstood that the mutated codon encoded the same protein instead of the same amino acid. So that's the idea. You need to be careful. And then for part C, if this type of mutation accumulates over time in the DNA molecules, there is a chance that it will affect the functioning of the protein formed and subsequently lead to tumor formation. And you need to suggest which cellular process this protein controls. So the idea is that because if you keep eating the grilled meat, so you may uptake a lot of the carcinogenic chemicals. And these chemicals, they will randomly attach to the guanine to make it misread as thymine. This mutation will take place in the whole DNA strand. So you will know that there should be a lot of guanine in the DNA strand, right? So there will be a different site of the mutation. And then one day, this mutation really happened in the particular region for the coding, and other type of amino acid, and it will really affect the conformation of the protein or the function of the protein or the active site of the enzyme molecules. So there will be the problem. So the concept checking is that we need to identify the cellular process which is not well controlled, so may lead to cancer or tumor formation. And the thinking logic is that we have the genes to control the particular protein synthesis, and this protein can control the cellular process, and this cellular process obviously is the cell division. Cell division is normally controlled by segments of the DNA, it means the genes, in the cells. However, when mutation occurs in the genes, the cell division may become overstimulated or may never be inhibited. So this will produce a mass of abnormal cells called tumor. So that's the tumor formation mechanism. Therefore, the possible cellular processes can be control of the cell division, control of the mitotic cell division, or control of the cell cycle, or control of the DNA repair. So let's talk about the curriculum mapping. First of all, for the base sequence, and then we talk about the protein synthesis and the mutation. Firstly, we need to introduce the mutagen. This time, the carcinogenic chemical is commonly found in the grilled meat. So maybe next time the question will ask you that, what about we do not grill the meat? We boil it and we steam it. Is it a method to lower the chance for us to uptake the carcinogenic chemicals? Surely yes, because for boiling, for steaming, it's not that high temperature because it's just 100 degrees Celsius, right? But what about for grill or barbecue? So we may use 200 or 300 degrees Celsius to cook the food. So it increases the chance to produce the carcinogenic chemicals in the food. And then we talk about the base sequence and the mutation. So we have the idea about the gene mutation, deletion, insertion, substitution, and inversion. And apart from the gene mutation, we have the chromosomal mutation, for example, Down syndrome, or with one more or less sex chromosome. And then for the protein synthesis, we need to recall the idea about the transcription, translation, and for the codon feature. This time we talk about the degenerate codon, but what about trilap codon, non-overlapping codon, and the universal codon? What does it mean? You need to be familiar with that. And then we combine all the idea. We have the tumor formation. And that's the idea we learned it from the basic genetics and the molecular genetics. The gene, it can instruct, it can direct the protein synthesis. And then the protein, they are involved in the metabolic activity or the cellular processes. So what if there are mutations in genes or the chromosome? it could be something wrong, lead to tumor formation or cancer formation. So for the tumor formation, it recalls the idea about the cell division, cell cycle. And for the tumor itself, we talk about the benign tumor or the malignant tumor. So before that, we have the questions about the mutation and also the cancer. So watch the video and do revision. And lastly, we talk about the possible question variation. Does a deletion or insertion of a single base result in a change of the amino acid? Explain your answer. Secondly, does the continuous cell division of the tumor cells result from a shortened interface, or is it due to an accelerated mitotic cell division? It means that the interface shorter of the cell at rest stage, it becomes shorter and shorter. Or for the prophase, metaphase, anaphase, telophase, cytokinesis, these processes, they keep speeding up. So tell me what you think in the comment section.